I would like to know, how do you dispose of the completely misfolded proteins?
Well, this is a very interesting question because there's no clear answer for that. There's a complex of protein complex composed of several components, and the composition of that complex depends on which, where is the misfolding, the cause of the misfolding in the protein. For instance, if it's in the membrane domain of the glycoprotein, or is it in the cytosolic domain, if it is a transmembrane protein, or in the ER domain. But all of them, I mean, if it's in the ER domain, it has this, one of the components is this lectin, OS9, that specifically recognizes the exposure of this alpha-1,6 linked mannose that is exposed upon removal of the two mannoses. And then this complex, it has a very weird composition. It has ubiquitin conjugating enzymes, ubiquitin ligases. It has ATPases. And actually, the translocon, the pore by which the misfolded protein is transported from the ER to the cytosol is unknown. I mean, it's
And are there any examples of proteins that must go through several cycles of glycosylation before folding? Or is it just a matter of luck that you get folded and then not glycosylated? Or how many times does a protein get reglycosylated before getting out?
The ER is a very plastic subcellular site, so there are many chaperones of different family, classical chaperones that help for the folding process. For instance, this glucosyl transferase is essential for in whole animals. I mean, if you knock out the enzyme, the animal is embryonically lethal. I mean, but in cells, you can isolate cells from the embryo, and they grow perfectly well. So that also, for instance, in yeast, you have to apply very high stress to the ER, high ER stress, to find that the glucosyl transferase is essential. If not, the cell can manage without this calnexin calreticulin cycle. That indicates there are very few proteins that absolutely require this cycle of calnexin calreticulin cycle for survival.
Do you have more questions?
Yeah.
Rech?
Very good talk, thanks very much. A very, very interesting topic. I'm going to ask you, and I'd like your comment, in contrary to what Eddie just asked, relating the possibility for the cell fold the protein, a non-glycosylated protein, if you do have a high expression level of messenger RNA. We've got some results where you, after purification, you can get fractions glycosylated and fractions non-glycosylated. This is a very interesting and certainly a complex. I'd like your comments on that.
Well, that depends on the efficiency of the cell type you are working with. Glycosylation is a very precise process. It occurs when the SEC1 is an asparagine X serine or threonine. It's about 12 to 13 amino acids from the inner surface of the ER. If there are too many proteins being synthesized, you will skip the glycosylation process. I mean, that's
Bustamante?
Very nice talk. Thank you, Armando. You mentioned that this glycosylation occurs even in the presence of a molten globule.
In
did
the
I-
presence, yeah.
In the presence of the molten globule. And that it recognizes hydrophobic
Yeah,
exposed.
patches or
Right. My prejudice about molten globule is that it's a molecule in which the hydrophobic collapse has already occurred,
Yeah.
and that what has not happened yet is the organization of the tertiary contacts that makes the protein ultimately click into the native structure. So, what these results, what your results seem to indicate is that in this molten globule, which is already after the hydrophobic collapse, is still accessible. It's still fluctuating between exposing and non-exposing hydrophobic results. Could you comment about this?
Well, it's not a yes or no situation. I mean, you can get also glycosylation, glucosylation with the glycan linked to a rather short peptide, but very rich in hydrophobic amino acids. So it's not that you have to have a patch or it's just some hydrophobicity close to the glycan. It has to be rather close. I mean, it cannot be quite far. I mean, it has to be rather close to the I don't know how it works. I mean, it's what I said. The guys who solved the structures say that one of the thioredoxin subunits exposes hydrophobic surface, and that must recognize something and activate the glucosyl transferase that is at the end of the arc. So I really don't know how it works. I mean, it's
Yeah, just a historical note, but Leloir, who was a member of this academy, got the Nobel Prize for the discovery of UDPG, which works on the cytoplasm. Now we have another function for UDPG inside the endoplasmic reticulum. I wonder how it gets there, but still, Mandy was Leloir's select student, so I'm sure he would be very happy that you're still working at it.
That's just a point. The UDP glucose, the transporter for the sugar nucleotides have a very particular structure, and we knocked down, was studied by a very good friend, Carlo Hirschberg, and we knocked down in yeast all the possible transporter for UDP glucose. You knock down all the possible transporter for UDP glucose, and the glucosylation is still went on. So I think UDP glucose is transported to the lumen of the ER by different mechanisms, different from the antiporter one described for all other sugar nucleotides.
Well, on this note, we will break for lunch, and I thank all the speakers for the very high level of Latin American science shown here. Thank you very much.